All right, so this is junction dysfunction here. Been mapping out all the different mechanisms that I see. So what I like to do for a living is create databases. And I read a ton of papers and I can see a lot of different patterns, and I like to find universals, and I like to zoom in and see the tiny little things and details at the same time. So what we've got here is essentially a complex. So a complex is essentially what, let me go back to my face first. A complex is something that is able to diversify and turn into multiple things at the same time and integrate at the same time. So for example, take your hand. This is a complex. You've got individual fingers, right? Or they're all diversified because you can move them differently, but you can do something like grab something with it, or you can use it as one thing. The whole body works that way. The immune system works that way, along with the detoxification enzymes, they work that way. So many different parts of the body work this way as a complexification system. So let's go back to this here. So when we look in here, this diagram here, it's about universals. All right? Because there's no such thing as causal patterns, Like when we look at A cause B, like if you push a pencil, was it really that you pushed the pencil on a table and it rolled, or was it the physics of it happening? There's a lot to that, and I'm not trying to get super in depth in that kind of concept, but just get that into your mind when you start thinking about how the body works. All right, so this diagram is going to show universals. It's not a direct causal pattern, as we just discussed. This is a complex that differentiates and it integrates. And on top of that, it's able to self-reorganize, which means it can grow itself and kill itself and all that kind of thing. Like if you look at a tree, does it start off as a tree, or how does that work? Because it gets rays from the sun in the chlorophyll, and that gives it energy, but it also uses that energy to grow itself to get more energy and continues that pattern. Same thing like chicken and the egg. And this self-reorganization works on both positive and negative feedback loops. You'll see in a bit. And because of this simplistic layout that is on here, I'm purposely leaving out bioindividualities. Bioindividualities are the things that make everybody different, like your genetics or your lifestyle, or the things that the enzymes that you create, or the cofactors, your diet, the toxins and pollutants that you're exposed to on a day, your nutritional status, how old you are, or if you have any latent or hidden infections. Those are all bioindividualities, okay? So this is all a complexification system. It self-reorganizes, and we're going to look at the universals and not so much the bioindividualities. All right, so I'm drawing it out so it can help you see where people fall, and it can help me know when I work with my clients which parts need support, upregulation, downregulation, whatever, okay? So this concept of junction dysfunction, what is it? Junction dysfunction is essentially when your epithelial cells here and your endothelial cells. So we're just going to draw the endothelial system here. So the endothelial system is the blood, right? Blood travels through it. Well, what happens here is when these things break apart and it becomes leaky, so things can go through. This is junction dysfunction. So these junctions here aren't working. This happens in the gut. So we look at like the gut. And we've got tight junctions here like occludins and things, cadherins, and then those break open, and food byproducts can get in, or bacteria could get in. Parts of them can get in, activating the immune system. The blood's right behind here. So this whole concept of junction dysfunction really plays a universal system between anywhere in like the gut, the blood, the blood-brain barrier, the kidney barrier, the liver barrier, the heart barrier. There's just so many different parts to it. Mainly, we're going to be talking about things like, zoom in here, the endothelial cells. All right. We'll be talking about that. We're going to be talking about epithelial cells. We're also going to be talking about enterocytes. Let me back up. So blood vessels here, lung cells, the things that are exposed to the world, like your nasal cavity, your gut cells, enterocytes, and the glia. So your blood-brain barrier and the different brain cells that are in there that hold those neurons together. Okay? Let's first talk about the endothelial cells because the latest virus is very prominent in affecting the vascular system. So the endothelial cells, they make up all of your blood vessels of your entire body. You have things that go from your arterioles or your arteries, which get oxygen from your lungs and pump through your heart. And they bring those to arterioles, and those arterioles bring those to the capillaries, and those capillaries go to microcapillaries. Those microcapillaries go to venules after they've been deoxygenated and given glucose to the necessary cell that they need to go to. Then they go back up veins, which are able to dilate and constrict and bring that all back up to the lungs so you can excrete carbon dioxide. That way, that cycle all happens. This is called respiration. All right. So the endothelial cells. If you watched my last video, you would have seen that glycocalyx loss is a big problem. And if you didn't, let me just draw it out for you real quick. So on the endothelium, this is a blood vessel. We have blood cells that go through it. They're going that way. Let me draw that a little better. So that they're going that way. We have these little tiny hairs here called the glycocalyx. And they rest on top of all these cells. This glycocalyx is literally everywhere in the body. Not every single where, but it's in so many different systems that it's highly important. And the glycocalyx isn't known about a lot. Many people don't know about the glycocalyx because it wasn't able to be imaged in humans for at least the past decade. So it was just the past few years that research has been coming out about it, and it's an entirely different system. Now, this glycocalyx, it has a negative charge to it, and the cholesterol sulfate that's on these red blood cells, these little guys here, they also have a negative charge to them. And because the negative and negative work together, they're able to push that red blood cell through nice and swiftly. It's kind of like the glycocalyx is like a jelly layer is the best way to describe it. Some people describe it as the trees of the extracellular matrix. The extracellular matrix is this system here that if you look at a cell, it has all these receptors on it, right? That's able to bind to things, whatever protein is coming in, so it can bind to there. Well, this glycocalyx, this hair-like structure, makes up like this larger system here. So like this tree-like system. So they call it like a tree in the forest, and the receptors and all the other things in the extracellular matrix that make up this other part, the receptors and all the binding actions, it's like the grass. That's pretty much how it's described usually. So when there's destruction of the glycocalyx, oops, I didn't mean to erase that much. Okay, whatever. When there's destruction of the glycocalyx, you get a positive charge here. And so this cholesterol sulfate here, this red blood cell is going to bind to here. So it's going to get stuck there. All right. Not only that, it causes this whole junction dysfunction stuff to get started. This is just one mechanism that the junction dysfunction stuff happens. And things that are in the blood get into the tissue and they're not meant to. This is actually generally not meant to, but in inflammation, it's meant to do that because you want immune cells to be able to get in here and fight a pathogen. You know, you want them to get in there, and if there's an infection in the tissue, you want them to be able to get there. That's important. What happens with these chronic post-viral illnesses is that this stays open. And so what you get is called, draw this out here, systemic capillary leak syndrome. All right. I like to call it transient systemic capillary leak syndrome because it's not the same thing as being on your deathbed. So when you study this in the textbooks, just systemic capillary leak syndrome, it's when people are literally like bleeding out onto their skin and all that stuff. But if you've had long COVID, let's just say that, or COVID, and you have long COVID symptoms, like some people will notice that they have blood pooling or blotching or weird dots of like deoxygenated blood on their hands, right? This is essentially what's happening here. Not only that, you also get, pull up over here, another thing called sepsis. And sepsis is essentially when bacteria are able to get into the tissue or into the vascular system. I like to call this more on a level of microsepsis because it's not full-blown sepsis because yet again, you're not on your deathbed in the hospital. So the combination of these two create this thing called junction dysfunction. All right. Microsepsis makes you very sensitive to all the different pathogens you pick up. You get so many different secondary infections. All right. And then that leads to this junction dysfunction. So transient systemic capillary leak syndrome plus microsepsis equals junction dysfunction. All right. Let me just check with the chat that everybody is able to follow along here. Hello from Paris and hello from Vienna. Nice to see you too. Okay. So when we pull back, we're looking at the big picture here, right? A lot of people like to focus, a lot of people are trying to just hyper focus on the mechanisms, but hyper focusing on the mechanisms doesn't allow you to see the big picture, right? Like if you were trying to understand what a tree was, but you were only looking at the pieces of the bark, or you were only looking at the leaf, or you're only looking at the root, or you're only looking at the mushroom that was growing on it, you would never get the ability to fully understand what you're seeing there. You have to take a step back and be like, oh, this is a tree. That's a problem with modern healthcare, is that we have many specialists that focus on just one part of that instead of looking at the entire picture. None of them are taking that step back to try to see the whole thing. And what's really helped me understand this kind of stuff is reading so much and then always trying to take a step back and seeing, how does this apply? You know, because I had long EBV back in 2012, or I got it over my summer between transitions of sophomore and junior year. And I was not able to experience the rest of my college career because of it. I was just bedbound. I was tired all the time, constantly getting sick. And so since then, I've been consistently studying because all the doctors I saw were giving me prescriptions for SSRIs. I should go see cognitive behavioral therapists. I tried them all. They didn't help. So I had to take it in my own hands, and this is over a decade later worth of work of understanding post-viral illness, okay? So let's get into the glycocalyx of the endothelial cells. When that breaks apart, I'm going to frame this more into the frames of SARS-CoV-2, okay? When the glycocalyx gets broken, it allows the receptors, the ACE2 receptors, the ones that SARS-CoV-2 binds to bind to and get into the nucleus and transcribe, essentially. When it binds to ACE2, that causes ACE2 loss on the cell. So the cell loses its ACE2 receptor. It essentially goes bye-bye. And this causes a problem with the renin-angiotensin system, the RAS system, RAS, for angiotensin 2 to bind to angiotensin 2 type 1 receptors. And what angiotensin 2 type 1 receptors do is they cause vasoconstriction, so constriction of the blood vessels. They cause inflammation. They cause clotting issues. A lot of the things that you see people getting with long-term consequences of COVID, right? And normally, when ACE2 is supposed to work, ACE2 is supposed to be able to convert angiotensin, angiotensin, let me try this a little bit better, angiotensin 2 into angiotensin, oh my goodness, my dyslexia is acting up. Angiotensin 1-7. This will bind to the angiotensin 1 receptor. This is able to bind to the angiotensin 2 receptor. So the angiotensin 1 receptor here is inflammatory, all right? While the angiotensin 2 receptor has anti-inflammatory effects. It helps with vasodilation, helps with your ability to breathe, so you can open up your airways. When this is activated, your lungs become constricted. A lot of people say after they've gotten COVID, they've got chronic asthma, and all the steroid sprays for asthmatics don't really help, right? It's because this ACE2 enzyme is not able to work anymore because you've lost this receptor. And so in the blood, let's go back to the blood, you have free-floating ACE2 receptor, and it's just flowing around. You know, you've lost that ability to create this pathway. This is a big role with what we're dealing with the ongoing pandemic. So you get soluble ACE2 that activates angiotensin 1. Like we said before, that causes inflammation and also vasoconstriction. So your ability to breathe becomes a lot harder. Your veins seem to be like popping out and smaller at the same time. Like you've got a lot of blue veins on you. Angiotensin 1 receptor activation also causes platelet recruitment. So platelets are essentially these cells. They don't duplicate themselves. They come from bone marrow, essentially. They don't have a nucleus or anything like that. What they're able to do is they're like the conductor of an orchestra of wound healing. You know, they'll patch up leaky skin. Like if you cut your hand, platelets are there to save the day, you know? That causes adrenergic activation, fibrin, and clotting, which is good. This fibrin clotting part is good, but it's too much because you've lost this up here. And then you get agglutination. Agglutination is essentially when red blood cells get like stuck up on top of each other. So if you've ever seen blood, they get like, you know, they make these like big things. And they can also connect with pieces of fibrin, making like this like thrombosis, like kind of big clot here. And imagine that going through blood vessels. That's like very difficult to get through a blood vessel. You know, it's going to get stuck on parts where you have glycocalyx loss. What this causes is thick blood, like thick blood. You know, if you ever seen, a lot of people are prescribed proteolytic enzymes, right? Serratiopeptidase, lumbrokinase, all those kind of things. They're trying to break down this fibrin here, cut down that fibrin. That way, these cells can, you know, be a little bit more free, but that doesn't address a lot of these problems that we're going to talk about for the rest of these things. So as you would have guessed, if the blood cells get cut off here, that causes thrombosis, a thrombotic event, lack of blood to an area, right? This ends up in autonomic POTS, and we'll have to discuss autonomic POTS a little bit later because it has a whole mechanism for this that I've written out, and I'm going to share with you guys soon. Okay. Also, when you lose the glycocalyx, let's see. Let's go back to, because we're still talking about angiotensin 1 receptors. So those leaky junctions, which we were talking about over here, right? These leaky junctions, they continue to cause angiotensin 1 receptor activation because it's a positive feedback loop to try to heal itself, right? Like we talked, there's a complexification system. With that, you get microcapillary loss. Now, microcapillaries are this system. So if you saw my last video, you might have not. This is what it looks like going into your tissue, how blood is able to transmit glucose and oxygen and all that fun stuff. Okay, so behind here is a bunch of tissue. Let me just see if I can get a big thing on here. This is all tissue. Yeah, there we go. This is all tissue in here. And then inside of here, you have these red blood cells that are traveling this way, right? These here would be called reserve microcapillaries. So this would be a reserve microcapillary. I'll zoom in here a bit. This would be a reserve microcapillary. Same thing with this is a reserve microcapillary. And what reserve microcapillaries do is allow you to adapt to different vasoconstrictive or vasodilative events. So it helps you vasoadapt. So if you're laying down flat, you're not really using your vasoadaptive system. So your blood will just travel down one of these microcapillaries or just a couple of them. Now, when you stand up and you start moving around, walk somewhere, jump up and down, whatever it is, it requires more blood to be going through here because you vasoconstrict when you stand up. You know, your legs are supposed to vasoconstrict. That's why like POTS patients will have to wear compression socks because they can't vasoconstrict properly. We'll get about that. That's a lot of these different things here. But when you're healthy, they're able to go down all these different channels. That way you can have multiple red blood cells that are coming through here, able to get to all the different tissues. And they come out through on the other side. They deliver their nutrients to the tissue where they need to go, like glucose and oxygen. And when they come out of here, they are, I need to pull out a different color here. There we go. Deoxygenated. So when they come out of here, they're deoxygenated, right? They don't have oxygen or glucose and they look blue. You know, like when you cut yourself and you look at your skin, it's like blue, and then you cut yourself and it comes out red, it's becoming oxygenated. All right. And then that goes back up to the heart and the lungs to get more nutrients. Now, what happens when you lose that glycocalyx layer? Let's get a good color here. When you lose that glycocalyx layer, let's say you got a glycocalyx at least on one of these capillaries, it's able to repel that red blood cell so it can travel through appropriately. But if you lose it on these channels, you get essentially blockage of these systems. So these red blood cells are no longer able to go through there. So when these red blood cells are going through here, they're going much, much faster. Much, much faster. Let me see if I could pull up a photo for you to show what this looks like in a study. Here we go. So on the top left here, you have your reserve capillaries, right? They're not being used because you're at rest. But when you do a vasoadaptive thing, that causes it to use the other microcapillary channels. Now in sepsis, this is a model of sepsis. This is why no one's talking about it in COVID or any viral illnesses, because they can't link the two because they hyperfocus. Doctors like to hyperfocus on just their profession and not think outside the box. So this is the left here, sepsis at homeostasis, or not doing a vasoadaptive event. And then we have on the right here, when there is a vasoadaptive event. So essentially, instead of getting these reserve channels to disperse the blood is shooting much faster at homeostatic rate, like if you're laying down. So your heart will be beating normal than it should. So your heart rate variability will be a lot lower just at rest. And when you try to do a vasoadaptive thing like stand up or go into a hot shower or sauna or whatever, you probably couldn't even do sauna at this point if you're that sick. Those red blood cells would shoot through so much faster. And so you get tachycardia, and tachycardia can lead you to pass out. That's essentially what vascular POTS is all about, right? When this happens, you essentially get hypoxia to all the tissue in here. These tissue need oxygen and glucose to work, and these go all from anaerobic, which means that they all go from aerobic, aerobic respiration, all the cells in here, to anaerobic respiration. Man, drawing with a mouse is not the most fun thing. You get anaerobic respiration, and anaerobic respiration comes with downstream byproducts like hypoxia, inducible factors, and mitochondrial dysfunction, and a whole bunch of other things that we're going to get into. So this is a big system here, and it's a universal. It's complex, and it's diverse, and it integrates. All right, so let's go back to where we were with this stuff here. So we're back at the leaky junctions, microcapillary loss. That microcapillary loss causes the vasoadaptive loss. So you can see now how you're unable to constrict and vasodilate and respond appropriately to those adaptations. And that's what causes vascular POTS. You'll see in a decade or two, doctors will finally start to be talking about this, because microbiome research, you know how hot that is right now and how many doctors are talking about it. When I saw a doctor 10 years ago and I was asking him about like, oh, does my microbiome play a role in my post-viral illness or my EBV? First of all, they told you can't have lung EBV, which is total BS. And second of all, they said, no, the microbiome doesn't exist. Like, your gut is sterile. Or some people have it in very rare circumstances. And now we all know that's completely false. Anyway, so you get hypoxia. Hypoxia is going to lead to anaerobic mitochondrial respiration. That creates things like carbon dioxide, lactate, and hydrogen, which gives you shortness of breath and post-exertional malaise. I hope you're linking some connections here, and this is insightful for you. This anaerobic mitochondrial respiration also creates a lot of reactive oxygen species. Reactive oxygen species are essentially just oxidative stress that are highly, the term is reactive, but it's bad to describe something with the same name. So they are very excitable. They can connect, they can damage things right away. That's what it means by reactive. Reactive oxygen species. And that causes what's called autoinflammation. So if you have an auto immune disease, that's part of this process. So auto means that your body's attacking it, and inflammatory means that it has an inflammatory response. Okay? And then the other channel of this is getting mast cell issues. And that causes issues with vasodilative granules that mast cells secrete, and that also leads back to vascular POTS. It feeds back on this cycle here, you know, like it's going to go back up here. Also in the endothelial system, you get a stuck delayed immune cell response or activity. This creates a lot of immune senescence. Senescence is essentially zombie cells. Like when a cell doesn't self-destruct, that's because let's take a look at the endothelial system again. I'm gonna draw this like 20 times until it gets in your head. And then we got that microcapillary loss, like at one of these parts here. Now we got an immune cell, and it gets, I need more colors, don't I? And it gets stuck here against the endothelial cell. And so they're not doing their job. They're not getting into the tissue. They're not doing great things. And they kind of just hang out there like chilling because they don't have the charge for it. This creates a lot of viral debris as well, this delayed immune response. And you get a mismanagement of your immune system. You have multiple branches of how T helper cells can differentiate, and one of them is with Th17. Th17 promotes the immune response that leads to autoimmunity. That leads to that. You also get a whole bunch of other things like Rantes. Rantes causes things like more inflammation and hypoxia, nuclear factor kappa beta, hypoxia inducible factor 1 alpha and beta, and 2 alpha, and growth factor beta 1. You get things like glycosaminoglycan loss, which makes up that hair-like structure. You know, there's a lot of different things that make up the glycocalyx, and one of them is a glycosaminoglycan, and that happens via metallomatrix proteins. And they'll cut these things like a pair of scissors. And they'll go chop. And you get caspases. Caspases lead to more inflammation. What this NF kappa beta also, yeah, so it leads to the MMPs that feeds into this whole system back here. You also get hypoxia, which you can guess what hypoxia does over here. Well, it also messes with your ability to regulate glucose and insulin, and your ability to remodel your endothelial cells well, which further contributes to the junction dysfunction. Let's zoom out a little bit. Got a pretty big board here. All right. Now, still in the endothelial system, when you lose the glycocalyx houses a lot of different things, like we said. It's like those trees. You lose these, you have something called superoxide dismutase, SOD. And SOD is able to take, it's two oxygen molecules. It's called superoxide. And superoxide is another free radical, just like reactive oxygen species. And reactive oxygen species, or sorry, superoxide, when it combines with peroxynitrate, or sorry, when superoxide combines with nitric oxide, this creates an even stronger free radical destroying enzyme called peroxynitrate. You also might have heard it as UNU. I love that word. It's so funny. UNU. Okay, anyway, so because you have all this superoxide and the glycocalyx houses the SOD2 or the SOD, you lose a lot of the ability to house that SOD. The part that makes the glycocalyx like jelly kind of is this SOD, superoxide dismutase. This causes a problem in tetrahydrobiopterin in your endothelial nitric oxide system, and essentially upregulates oxidation. This system here is why it's a bad idea to take things like arginine or citrulline when you have loss of the glycocalyx and you have too high of a redox load. Because it's just feeding into the system, you're creating more nitric oxide. And when you have a lot of superoxide, you're creating a lot of peroxynitrate. This is why people do awful with with nitric oxide boosters. They're damaging themselves without getting the system all built back up. So like we said, that creates reactive nitrogen species and peroxynitrate. That affects your system, your redox balance. Your redox system is essentially your balance between the antioxidants in your system and the oxidants in your system. You want a healthy balance because they all do different things. Like you need oxidation for certain transcriptions to turn on in genes, but you also need antioxidants to turn those things off or they continue running. This is like where you get epigenetic changes. You get overactivation of the detoxification system, and you lose your ability to, or you increase your CYP enzymes. CYP enzymes are part of the phase 1 of your detoxification. And their job is to make something, a toxin, create an oxidative response of a toxin. That way your gene can turn on to start to go to phase 2. And phase 2 is all about making that toxin more water soluble by reducing it or adding antioxidants to it. Anyway, that causes poor detoxification. So you lose a lot of your ability to create sulfur groups, or you use up your sulfur groups and acyl groups and methyl groups, and your glucuronidation can go off, your quinone and you get epoxide depletion. Essentially, your detoxification is like using up all these groups to fix this redox imbalance, right? This makes you hypersensitive to stressful things that weren't even like that stressful. Like it could be mental things, it could be toxins. Like plastic is a stress on your body. Like microplastics are a stress on your body, or smoke is a stress on your lungs. Whatever, right? That makes your hypothalamus and pituitary adrenal axis go out of whack, producing a lot of corticotropin releasing hormone, cortisol, feeds back. It's a positive feedback loop. And that causes impairment of your limbic system, essentially the cingulate gyrus, which regulates emotion, and the hypothalamus, which is able to regulate your autonomic nervous system, and which talks to the pituitary, your hippocampus, which is able to form memories and recall memories, and your amygdala, which is like that fear center. That also leads to adrenergic activation, and you get autonomic POTS. All right? Are you starting to see some trends or similarities here? Okay. So with poor detoxification, it's easy to absorb toxins because you're not detoxing them. You're not adding these groups to them. Like sulfur, when you add a sulfur group to, for example, vitamin D, the active form of vitamin D, just put D3 here, you get deactivated vitamin D. Like it's important that methyl groups. Like, do you hear how many people have like methylation issues? Like looked at before. I mean, the world is very toxic, and there's a need for all these processes to happen more often. But when you're sick and all this gets out of whack, like you go through these really crazy, like really fast. And so you have to support this and you have to look at your genes and you have to put all your SNPs into some kind of encoding website that's going to give you like if you have methylation issues or acetylation issues or whatever it is. Yeah? Okay. So you absorb toxins. Toxins are very immunosuppressive, as you can probably guess. That increases your ability for creating those autoinflammatory cytokines, the Th17 side, and your loses your immune tolerance side, which is your Treg side. And autoimmunity, inflammation, auto-oxidation. All right, so that's what I've drawn out so far for the endothelial side. There's a lot more to it, but I feel like it would just get too complex. Okay. Now let's talk about epithelial cells. Epithelial cells, those are the lining of, I need to drink something real quick. Hold on. So epithelial cells, they have villi on them or areoli on them, which make up your lungs. They make up like your nasal cavity, your mouth. Like all those are epithelial cells. When you lose the glycocalyx and you get you get junction dysfunction because of the systemic capillary leak, where are we? Here we go. Or microsepsis. Remember, microsepsis causes endotoxemia. If you've never heard that word before, we'll go through that in just a minute. And that happens from LPS, lipopolysaccharides, and a lot of other different things, like fungal byproducts as well. All right, so what we're talking about, glycocalyx loss, that causes, oh yeah, endotoxemia. I don't have to draw it out, it's just on the board. Endotoxemia makes you very sensitive to, this is why this is a first edition. There we go. Okay. But what we could do, and a good way of putting this is leads you sensitive to air pollutants like smoke and all that kind of stuff, right? Like there are people who can tolerate smoking away like and feel totally fine because they didn't have all this whole process happen. But there are other people who were totally fine smoking away and then they got sick and now every time they smoke their like lungs are like totally like collapsed. They got that bronchoconstriction and all those other issues. This also causes, so let's just write that out, air sensitivity, air quality sensitivities. Endotoxemia leads to toll-like receptor activation. Toll-like receptors are part of the innate immune system. They're able to signal or to identify pathogens. They're either able to identify pathogens or damage of the body. So you got PAMPs and DAMPs. Those are the funny terms. Right? That also causes microcapillary loss. And you get vasoadaptive loss, hypoxia again, mast cell issues, which also lead to vasodilative reactions. So there's a lot of trends here that this happens. This glycocalyx loss and junction dysfunction leads to secondary superinfections. Superinfections are essentially when you have multiple infections compounding on each other. Like people who have immune suppression from mold or whatever, they tend to have like issues with Lyme, like Borrelia, Babesia, Bartonella, all these different things. It's like, wait, like, did they just get infected with a whole bunch of diseases at once? No, because most people have those pathogens in their system. It's just their immune system is able to keep it in check, right? Secondary superinfections also lead to the toll-like receptor activation. Toll-like receptor activation like will lead to things like interferons. I know a lot of people are studying just purely interferons, like that's the problem, and it's not. I mean, like it's a downstream product. Like if you hyperfocus on something, you can make anything the problem. Microcapillary loss, vasoadaptive loss, hypoxia, mast cell issues, vasodilative reaction. I really don't believe in mast cell activation syndrome. My mentor, 8 years ago, was Dr. Dr. Theodoros Theoharides. He's the mast cell master. He has the most literature about mast cells, especially in relation to autism and all these like weird complex things. Um, and he really sparked my interest in getting the mast cells. But when I was studying them, I was like, wait a minute, these aren't a part of the problem. These are actually part of the solution. You know, the body's trying to heal itself. Your body wants to heal itself. It wants to heal itself. It's you know, let's just throw a drug at it. All right, another part of the junction dysfunction is the gut. So we got the enterocytes. When we look at the enterocytes, I'm just going to draw like a enterocyte here. That look pretty cool. Oh man, that's the best drawing I've done yet. I went very slow with the mouse. They have these little things here as well. Guess what those are called? Yeah, they're glycosaminoglycans and all those fun jazz. They make up the glycocalyx. So you can imagine what happens when this stuff gets damaged. Oh no, how do I make this much smaller? There we go. There we go. I get to delete little things at a time. All right. Oh no. Okay. So when that happens between the enterocytes, let's just pretend this is another enterocyte. I'm just not drawing it. Those tight junctions, those were holding them together, get damaged. And so like zonulin gets upregulated. And you might have heard of like leaky gut. This is what leaky gut is. Intestinal permeability and like things like gluten, they get in there nice and easy. They activate the innate immune system, those toll-like receptors, and then gluten can also get past it and get into like places like, what's a good color for this? Your thyroid. Yeah, I'm not really going to draw it out. Your thyroid and your neck. You know, like how many people do you know have autoimmune thyroiditis? All those kind of things. There's the Adam's apple. Nice smiling person here. Not knowing that their thyroid's being destroyed by eating that wheat bun because of this whole thing. This will lead to, let's talk about what happens. This leads to loss of your ability to absorb fat soluble vitamins. So a lot of people will have issues with absorbing vitamin D, especially if they have like gluten issues or thyroid issues. And a lot of people try to relate it to like, oh, vitamin D is actually really good for autoimmunity. And it's like, yeah, it does work on a two-way street, but like, or it does work on that way, but it's a two-way street because the reason you don't have high vitamin D levels is because you're not able to absorb it through the gut. Like if we take a look at a gut cell, I'm gonna draw another one here, just really quickly. Cool. You have in here, which is called the lymph system, and you got it essentially hangs out like this, and the lymph system's able to like get rid of debris, but it's also able to absorb fat soluble vitamins. So like let's say like vitamin E comes in over here, it normally will like be able to be absorbed there. But if you've got this whole loss, there's a whole bunch of inflammation here. It's not able to get into here. So you're not able to absorb the vitamin E, and this pathway is blocked. Back to this, not only does big gluten come in here, you got bacteria that hang out over here as well. Bacteria, you know, they're part of your microbiome. They're good. Bacteria, most of your bacteria are just meant to be in your body, unless you picked it up from the external world, like a bad bacteria. Those bacteria are able to do something what's called pleomorphism. Pleomorphism is the ability for them to change their ability to become what's useful for you. Like humans only have so many genes, and we're only so adaptable. But bacteria, they have this amazing thing called horizontal gene transfer, which allows them to transfer genes between each other. I mean, they have a whole communication system, but this horizontal gene transfer allows them to turn from like good bacteria. Look how cute that little good bacteria is, to like a what's a good color for this? Good bacteria into angry, bad bacteria. And then you get dysbiosis because this balance here is off. You got a bad balance between these two guys. All right. Dysbiosis, endotoxemia. Endotoxemia is essentially when the pieces of the bacteria, whether it's from good or bad, are able to get into here and activate these toll-like receptors. All right, let's just check in with everyone here because I don't want to be ignoring anyone in the live stream. I'm not looking at it right this second. Do. Barrier disruption. Yeah, we'll talk about the blood-brain barrier disruption for sure. I don't know if I've read that paper. I've read tons of papers about it though. Okay, thank you, MetaMind, Isabel. And yeah, what causes sepsis? Okay, yeah, I think I answered that. I don't have any. Oh, you don't have methylation issues with NutraHacker. All right, that's cool. There's other genes that can become an issue. Their transcriptions become off. Methylation isn't everything. Like my methylation is totally fine as well, but like I became sick, and so I had a lot of issues from that. All right, so let's get back to the drawing board here. The endotoxemia and the dysbiosis leads to people being more sensitive to the food you eat, food sensitivities, activates toll-like receptors. I mean, you can get interferon alpha or interferon gamma. This leads to disruption of your ability to handle tryptophan and convert it into what it needs to be in the body. So tryptophan normally gets converted into serotonin. And you've probably heard of serotonin as being like the happy hormone, which has been debunked. Hold on one second. I'm going to drink more. I live in the desert, so I've got to re-moisten my mouth all the time. So tryptophan normally is able to turn into a few different things. Serotonin, which is 5-HT. And 5-HT plays a role on the vagus nerve, which makes you feel like, calm, relaxed, it's able to help you promote that feeling of like rest and digest. You feel calm, that's like whatever. But what this causes when you lose this ability to create it in your gut, because your gut is your main storage area or your main reservoir of serotonin, because it does this tryptophan metabolism. And when that happens, you have a majority of your nervous system just hanging out around the gut because it has they're called gastro transmitters. And then those gastrotransmitters, they act on neurotransmitters. There's a whole feedback cycle. But when you've lost this 5-HT, you get sympathetic dominance. Sympathetic dominance is essentially a fight or flight, or fight, flight, freeze, or whatever you want to call it. And that feeds back on the limbic system. Like your limbic system becomes dysregulated. Not only that, we're gonna talk a few other things, but your ability to create melatonin is decreased. Melatonin is important for turning on clock genes that tell your body what time of day it is, especially at night. So you'll get insomnia and you'll get poor sleep. Not only that, let's add another thing here that I forgot to include. I'm just thinking it now. Uh, it would actually fall over somewhere like this. Thanks for being patient with me, everybody. So, can you detach, please? Thank you very much. Oh, all right. So we got poor sleep here. And it's not poor redox. I need to write another thing in here. So you get poor sleep. Your body is able to talk to the suprachiasmatic nucleus, which is the center in your brain. It's really tiny, but it's able to pick up on signals of light and other what are called Zeitgebers. It's the German word for biological cues. I didn't design that word. You know, there's so many words in science that like I don't know why they call them that. Like there's a pathway called the sonic hedgehog pathway. I mean, it makes me feel like a child when I read about it, but like, you know, it has a lot to do with cancer and other things like that. But it's hilarious. I feel like someone who was at a university, they were like up late, you know, playing Sonic Hedgehog on their Nintendo, and then came up with the idea of like, oh, we just discovered this new pathway. Let's call it Sonic the Hedgehog. So you get issues with your suprachiasmatic nucleus because it is a um, feedback mechanism. All right. Let's go back up to more about tryptophan dysregulation. That causes IDO to upregulate. IDO essentially creates kynuric acid, kynurenine, and quinolinic acid. Um, these are byproducts that are necessary byproducts in inflammation. Um, they so like uh, kynurenine is able to downregulate the expression of N-methyl deaspartate receptors. N-methyl aspartate receptors are receptors in your brain that respond to glutamate. Glutamate's like hyper-excitatory, and you need glutamate to function and create new memories. It like makes things feel like alive and all that stuff. You know, some inhibition of NMDAR is great. It's kind of like an adaptive mechanism of kynurenine, but too much of it creates the inability to retain new memories or info, and you're just kind of like brain fogged. Um, so like when you get too much NMDA from glutamate, it's highly oxidative, which in that of itself can cause issues with creating new memories. But hyper inhibition also leads to this. So like a good example of an NMDA antagonist, something that blocks that is magnesium, um, magnesium glycinate. Uh, but if you've ever taken too much of it, you'll notice your ability to like stay focused and whatever, like it kind of makes you tired. It's not just magnesium glycinate. I mean, there's other forms of magnesium, not magnesium oxide, but like other forms. So like if you've ever taken magnesium, like too much magnesium, and just been tired, that can be helpful if you're taking it before bed. But that inhibition, if you're trying to be awake during the day, it could be kind of difficult. Um, this also causes issues with your ability to create vitamin B3 isomers like NAD. Nicotinamide products have just been like super promoted for like longevity and all this kind of like fun jazz. Another trendy thing. uh, but when you lose the ability to have NAD, it is important for cellular respiration. So you get poor mitochondrial complex respiration. Essentially, the ability for your mitochondria to produce ATP is shunted. What's the color I have after this? If I have any colors after this, I'm just curious. What goes after light green, dark green? Can I paste that over here? Cool. Because I want to just write that in. This is a first edition. This is just me translating my notes to something that is legible and more easy to understand, right? Cool. Oh, it's not supposed to say this. Uh, Low ATP. Let's talk about the brain. Woohoo! A lot of people have been asking me about the brain. And like, when I started my biohack, once I fixed my post-viral illness issues, like I noticed I had such bad brain function, and all I did was focus on the brain. I worked for a couple clinics. One of them was focused on rehabilitating people with Parkinson's disease. And then another one was focused on Alzheimer's and other like toxic insults to the brain, like concussions and all that kind of stuff. Like I had multiple concussions, and I still have the effects of it. I actually have a craniocervical instability on my neck, so I'm getting injections in a couple weeks to improve the ligamentous tissue. Anyway, enough about me. Let's go back to this. All right, astroglial cells. So let's take a look at what the blood-brain barrier looks like. I think that will help with this. So you've got glia. They kind of look like this. And they sit on the opposite side of your enterocytes. Because they want to be able to identify, not enterocytes, endothelial cells. My bad. Misspoke there. So they want to be able to see what's going through the blood and what needs to be getting into the brain, right? So like, you want nutrients to be able to get into here, and so glia can work properly. Your brain can work properly, absorb nutrients, all that things like glucose, oxygen, all the things that your brain needs to be awake and alive. When you lose the glycocalyx layer, you probably guessed it. I'll just draw it for fun. So there's the glycocalyx, got it on here, whatever. But let's say you lost it on wherever it is here. These things open up. These tight junctions open up. Let's just imagine there was a cell here, or these guys separated, their tight junctions opened up, and now these glia cells are like super reactive to whatever's passing through here. So you get a, it's called an M1 response for glia. So they can either be in their M1 phase or M2 phase versus M2. Now, M1 is great for inflammation and creating an immune response. So it's like highly reactive. Let me just see if I can draw out like a, yeah, reactive. And M2 is more anti-inflammatory and calm. Now, glia, they go back and forth between these states because it's just natural adaptation. You need to have amino adaptation, just like you need to have vasoadaptation or whatever type of like redox adaptation. You need all these different adaptation systems because you're a reorganizing, self-regulating system, complexification system. All right. And yeah, this guy's able to dampen, or go into an antioxidant state. So let's just make him like nice and whole. So you lose that glycocalyx there. This sets you up. So we were just talking about the N1 glia here. This will increase activation of those glutamate receptors, those NMDA receptors like we were talking about before, which will create that oxidative stress load on those brain cells, giving you brain fog. It will also contribute to your pain threshold. Your pain threshold gets much lower, so you react to pain a lot easier. You also get an increase in fractalkines. Well, that's a whole different discussion. I don't think it's necessary for this, but fractalkines will also give you brain fog. The increased pain thing is not supposed to be there, so hold on, bear with me. And depressive. You'll also get what's the term? Startle response, a heightened startle response. Let me move this out of the way. Whatever. Don't even need it. So yeah. And then on this side, because pathogens can get into the blood, that's how COVID is able to get into all your different organ systems, like including your brain. It could also go through leukocytes through your lymph system and get into your brain that way. It could also travel through your nerves, through your vagus nerve and get in that way to your brain. A lot of different ways COVID can get into your brain. Anyway, what this causes here is these secondary infections cause what are called proteopathies. Proteopathies, your ribosomes, let's just draw like the Golgi apparatus. Your ribosomes, they transcribe RNA into what are called proteins. And your proteins, they have functions. I hope this is bringing you back to your limit of elements on this page. All right, I might need to like delete a few things. Canva's giving, the whiteboard system is telling me I'm drawing too much on here. So let's just wrap this up. So RNA creates proteins, but when you have other infections, it messes with the ability for RNA to self-check itself, and it creates misfolded proteins, essentially proteins that are not what they're supposed to be. And that's why you see like in brains of people with Alzheimer's and stuff, they have, I'm just removing some of the elements so this thing works, this whiteboard works. That's why you see people with Alzheimer's have things like amyloid plaque or tau tangles in the brain. It's kind of an adaptive response to pathogens, but it's also a maladaptive response. And that causes reduced neurotransmission, and you get the whole spectrum of dementia, depending on what part of the brain that's infected. Like, if it's in your frontal lobe, your executive functioning goes down. If it's in your limbic system, your ability to like regulate emotion goes down, or feel other people, empathy. Like there's a huge disconnect in empathy, and that's one mechanism. There's a lot of different mechanisms, but that's one of them. Your substantia nigra is also another part that houses a lot of dopamine receptors, and in Parkinson's, like if it happens there, you get issues with movement. Okay, did we cover everything? Okay. I mean, what feeds back on this whole thing, apart from the microsepsis and the transient capillary leak syndrome, are like, this isn't supposed to say the same thing here. This should be a better color. This is actually supposed to be a color. Let's make this one light blue. So these things will contribute to it, and there are responses to inflammation as well, but they all feed back on each other, right? These aren't like, inflammation isn't the root cause. Like inflammation will cause these things to be produced, but it's not a root cause. Because as you can see, I hope so, you can see that they all play back on each other. So now let's zoom out, because we were just zooming in. Like, we looked under the microscope and we saw the bacteria, but we didn't zoom out to see the entire thing. Oh, all the bacteria are working together in some way, or whatever it is, like we're hyper focusing on a keratinocyte on the skin. I just said that wrong, but whatever. But let's zoom out. Oh, it's actually a wound. It's not just like a little piece of thrombotic platelet. All right, so what I've done is I've created a guide, and I'm still working on it. It's the first edition. And I know a lot of people have been asking me for what I've been doing with my clients and ways to, I'm a biohacker, ways to biohack this. And essentially what I've done for my post-viral illness, along with improving my health significantly and my vascular system, is I've created a guide. Like I've got some amazing photos in this guide. I just want to go through them with you real quick. Some just these like really, I try to make it as interactive and legible and easy to understand. That way, because I know a lot of people have brain fog. I had brain fog too. Honestly, I wish someone would have explained this to me or at least tried to explain some of these mechanisms to me when I was sick. I wish I wasn't just left with a diagnosis of anxiety, which, screw that. Anyway, I've got a guide here. It's got like tons of different mechanisms to it. Let me just see if I can bring this in here. They're all high resolution photos in there. These are just samples because it's a live stream. But we got a lot of different things like how systemic capillary leak syndrome happens, or what remodeling looks like with cancer, what the endothelial system looks like with thrombosis, how vasoadaptation works, like hormones, how they affect it. You know, people have asked me like, what's the difference between females and males when they get post-viral illness? We've got different things about like how the angiotensin system can give you dysbiosis. Yeah, more on the angiotensin system. This is more about like the glymphatic system. We've got some amazing things about the glymphatic system. The glymphatic system is the lymph system in your brain that's able to help you detox your brain. It's pretty much the reason why you sleep. Apart from like melatonin, which creates that antioxidant response. Okay, we've got the, you know, microbiome and stuff, like just got tons and tons of things that like very visual, all research based. There's tons and tons of references. So I encourage you, because I'm only one person, I encourage you to like look on it and like critique it. And I am a true believer in getting feedback to improve. So I want to make this as helpful to you guys as possible. Like how bacteria can cause thrombosis. Really, really cool stuff. Like tight junctions here again. And other things about tight junctions, like, you know, you should be testing your albumin levels. I'm sure your doctor doesn't know how to accurately look at albumin. They just like look at your CBC or your comprehensive metabolic panel and is like, you're good. Yeah, like how cytokine storms with SARS-CoV-2, which is very different than what you've probably been told, unless you do the research. This is in the lungs. This is like very complex views of, how do I make this bigger? I can't make it bigger for you, can I? Like the glycocalyx and what it's looking like so many things, like the negative charge of the glycocalyx. The structures of the glycocalyx and vascular tone and how things are integrated into the system. You know, just so many different things. So what I've got for you here is essentially a guide here, and it's still in development, but it's got tons and tons of resources for you. Tons and tons. Essentially, if you go to mybiohack.com, let me just go there. And you use this code intro50 when you're doing it. You're going to get half off for this. And you log in, click find your biohack, go to sign up here. All right. If you need some clarification on instructions, there's a button here that shows you how to sign up with mobile or how to sign up with desktop. And once I go through this, we'll answer some questions that are in the chat. All right. You know, I've got like audio where I speak things in the layman that will really help you understand things, because I know this is very technical and complex, and I'm doing my best to try to make this like simple. So what you do is it will ask you, click sign up. It will ask you to create an account. And then once you created an account, you're going to click sign up again, essentially. And, you know, put in the code intro50, you'll get half off. And then when you want to start the program, you just, or the guide, essentially, you can go up here, click Junction Dysfunction, and it will bring you to, let me show you what it looks like. Here we go. So this is the whole map. I'm going to update this regularly. This is not a finalized product, but it has tons and tons of things that you can do to improve your health and post-viral illness, and working on these mechanisms and more, which I didn't even describe because, you know, my Canva project only allows me to put so many elements on a page. I might have to like upgrade to a different system to map this out in the future, but I'm constantly updating this. And the reason why you're getting half off, this is an introductory offer, is first of all, I thank you for watching this video, this live stream and participating in it. But, you know, So we'll come down. I've got most of this mapped out. The ones that are not bolded are not written yet. They are in my notes. I'm just working on translating them appropriately so it's easy to understand. Because I know this stuff is complex, and especially when you have brain fog or like you're fatigued and you're in bed, you just want someone to be like talking to you. And so there's audio with all these things. You know, I've been working on answering people's COVID questions that they've been asking me in Reddit groups, and I'm constantly expounding upon this. That way, people have the answers that they're seeking that they're not getting for their doctors. I mean, we've got additional resources and a ton of other things, but yeah. So I encourage you to if you have health issues, or if you're a practitioner and want to learn more about this kind of stuff, to sign up. This Intro 50, it will give you half off until the end of next Thursday. All right. So let's go over to the chat and see if there's any questions. Let me do this for the time being. Hold on. Just want to make sure you guys know what the code is. Okay. What do we got questions about here? Because there's so many individualities, it's like asking me right here in this live stream is not going to be helpful if you ask, how do I treat this? You know, I hope you can see how this is a complexification system. All right, so to the questions here. All right, what causes sepsis? Let's start from the beginning, because I did go. So I have read a bunch of papers. I haven't read the blood-brain barrier disruption and sustained systemic inflammation one, but would you be able to throw that link in the chat? Or send me, if you're on Reddit, send me a private message with that link. I could also just, I guess this is the quote of the title of the name. So if that's the case, I could also just look that up. Author said, for the first time, we have been able to show that the leaky blood vessels in the human brain in tandem with an hyperactive immune system may be key to drivers of brain fog. Yeah, welcome to last decade. All right. Not you, MetaMind. I'm talking about whoever wrote that paper. You want my perspective? Do you think autoantibodies and specifically targeting endothelial cells are the cause of it? You know, there are different types of autoantibodies. Some of them are to try to. I don't believe a lot of antibodies are there to hurt you. They're more as signals to tell your body, hey, there's something going on. Let's do something about it. But you get caught in positive feedback loops and negative feedback loops, and that keeps those autoantibodies going on. And you have to know the right techniques and methods on how to do that. And that's pretty much what I've laid out in this junction dysfunction guide here. What causes sepsis? Great question. The glycocalyx being broken down, or not even the glycocalyx being broken down, those tight junctions being broken down, or toxins causing pleomorphism, causing the toxins essentially create oxidative stress, which breaks those junctions down. I mean, you can have a part of your immune system that's trying to clean up the system, create a lot of like histamine and serotonin and prostaglandins that will essentially break those tight junctions, because the immune system wants to get in there and clean that stuff up. But you know, some people get past the point of no return where they don't they die. That's what sepsis on the deathbed is all about. It's unfortunate, and they give those people on their deathbed what are called vasopressors. If you've heard of like the drugs like losartan, like sartans, that's what that's all about. Big thanks for your information. I'm glad I'm able to help you, MS. Do you know why many long COVID sufferers are healthy on paper when measured by doctors, but have normal health parameters like blood pressure, oxygenation, saturation, heart rate? Yes, absolutely. It's because their testing methods are ancient. Their instruments for using for these things are very poor. Um, I really wish we would have the ability to do real-time proteomics, which is essentially the ability to look at proteins and folding and how things are happening in real time. I know it's kind of like a Star Trek fantasy kind of thing, but that's kind of where I'm hoping we get to in the future. That way, we're not looking at it like, your SpO2 could be totally fine, uh, but you could totally be burdened with carbon dioxide from those mitochondria and lactate, which gives you that feeling like you're hypoxic, right? Like your cells aren't getting that oxygen, but like, you're measuring your capillaries. Like your if you ever look at your eye and you can see those red lines going through, those are your capillaries. Your microcapillaries are smaller than that, and they need to be seen through a microscope. So like that's happening on like such a microscopic level that you can't even that those blood SpO2 monitors don't see that. I hope that answered that question for you. Can you have junction dysfunction but not showing it in the vital parameters? Absolutely. Um, Doctors are only looking for people on their deathbed. Their measurement ranges look at the general population, which, as you may know, the general population, the sample size, are mostly sick. And uh, they don't find optimal ranges. They're not like, who are the pro athletes or the people in optimal health that we're going to um, you know find what the best numbers are for in the male and female population. In fact, they only look at males. A majority of the biomarkers are just males. They don't even look at females. It's only in just the past decade that they look at females. So I mean, what I've been doing in my practice is, on top of working with people who have chronic health issues, that's probably about two-thirds, 75%, at least now with all this long COVID and post-viral illness stuff. That the other third, as well, I'm working on myself, is learning about what are the optimal levels for hormones and biomarkers and all these different things, and enzymatic functions with epigenetics and all that kind of fun jazz, for myself and with like, you know, pro athletes and people who are like really trying to optimize their health and aren't sick. So there's two spectrums that I've been working with, which has given me tremendous insight. Do do. Can you adventure? Okay, so I don't have any methylation issues to nitrihactor. Okay, that's fine. You want to look at other epigenetic changes if you're sick. Like what I've noticed is now that I don't have post-viral illnesses issues, that my ability to or my need for nutritional supplementation is like with methyl groups and acyl groups and all that kind of stuff. Like I don't need that as much. Like I can take a supplement holiday for a month and be fine. Um, but what I do is I really, really, really push myself in things because I like to really experience life. And so like sometimes I'll supplement with them. But once you get healthy, those epigenetic changes, they don't express. You don't need that stuff anymore. Is it wise to take vasoconstrictors when eating histamine-rich foods to counter vasodilation? That's a phenomenal question. So histamine acts on four different receptors: H1, H2, H3, and H4. H3 is my favorite because it enhances cognitive function, but that's not part of your question. H1 is a vasodilator. So when histamine binds to H1, it causes vasodilation. If it binds to H2, it causes vasoconstriction. So it would not be so much as taking a vasoconstrictor, but as taking a vasopressor. Looking into like the Sartan class. Um, can you give us some idea on treatments for your use? Yeah, the whole guide is about that kind of stuff. I would love your feedback about it. Wouldn't the junction dysfunction heal over time when initially damaged? No, generally not. Not without the right techniques. Like Tiger Woods had to go back to learn how to redo his swing before he became the pro of all time. I mean, he has his own problems, but he was like almost the best, and then he had to relearn the proper technique on how to do things before he became the best. Took him years to do that, but that's kind of the same. That's like an analogy for what I'm trying to say here. Why, under circumstances, should you be careful with nitric oxide boosters like L-citrulline and L-arginine? Great question. I tried to describe earlier, but essentially when you have a lot of when you have damage to the ability for endothelial nitric oxide synthase to work properly with its quenching ability of superoxide dismutase, so the thing that quenches the free radicals, any nitric oxide booster will make nitric oxide. But when it combines to superoxide, those two oxygen molecules, it creates that peroxynitrate, which is highly inflammatory and highly oxidative. So that's why I generally don't recommend it. Also, arginine itself puts viruses in their lytic cycle, which essentially they express, especially for like the herpes class viruses. I know a lot of people have latent viruses with herpes classes. Or hidden protein is still lingering. What's your take on viral persistence in general? You know, there's mixed evidence for whether there's viral persistence. I would say that there's low quality evidence to it, but I would believe that there are viral remnants, or at least presence in macrophages and leukocytes, and at least those I've seen them there. Yeah, but the guide should address that. What do you think of what do you think is causing, say, long COVID? Is the virus persisting, or is it autoimmune, or does it kick off junction dysfunction? I believe it kicks off junction dysfunction. You know, I think something that you'd really enjoy in this program is learning about, let me go back to here, is what this article here called, where is it? Retroviruses, hardware upgrades, and vaccines, latent viruses and immunosuppression, and asymptomatic long haulers. Those three will be very valuable to you regarding that question. Just pull up the questions again. All right. Thank you for all your research. My detoxification system appears to be messed up. You got high beta glucuronidase. Yeah, that's going to be an issue. Is the inability to detoxify the reason the lymphatic system gets so clogged up and slow in long COVID? It's one of the reasons. It definitely is one of the reasons. Yeah, you know, I'll make a further post. Like, this is the first edition, and I'm still building it out. But all these what is already posted on here is already super helpful and will get you miles ahead from whatever your doctor is helping you or trying to tell you to do. All right, guys. Any other questions, or I'm going to sign off. And remember, this code here is still usable till Thursday. You know, I want to give you the ability to get in early. Like, some really cool things in here. Like, we talk about the glycocalyx and the microcapillary system, the mitochondria and mast cells, and hypoxia, which is super important to know how hypoxia factors work, how blood clots and platelets and fibrin contribute to autonomic POTS, more things about bacteria that you may have never known. The whole thing on junction dysfunction, this might end up being 3 or 4 different sections, but that's kind of the order of what you should be reading this in. The RAS system, all about ACE2 and angiotensin 1 receptors, asymptomatic long haulers, some really neat stuff there. How the oral microbiome works and sulfur metabolism works, how to get negative ions, kind of like biohacking the system before your glycocalyx heals and those junctions are healed again through those multiple pathways. You know, different ways that COVID can get into the body, like through rage receptors. Or age receptors, receptors for advanced glycation end products, like so many different things. Like you need to know how to rebuild your gut. Like it houses so many different things, and tryptophan metabolism, like different ways to address the limbic system without gaslighting you. You know, like how vaccines work, latent viruses and immunosuppression, building that immune system back up so it works in a healthy way. How to get your sleep back in order and the melatonin working properly and all these different things. You know, extra tools, drug supplements, modalities that weren't discussed in all these other things. These are just extras. So they're synergistic adjuncts. More on genetics. So if you have like a epigenetic issue, we'll be talking about that. I'm still working on how to create this into a short summary because it's a lot. And yeah, so I mean, we've got tons of questions, like so many different things, like how does COVID cause aging, or how does the spike protein bind to the glycocalyx, or why do I have low cortisol levels? Um, how does why do you have cholesterol after that? Why do I feel better after doing a Myers cocktail? If you ever gotten one of those IVs, you know, there's just so many different things that people have been asking me over the past few years that I've had to find out why. I'm not one of those people who settle with like, oh, the research will just come out eventually, like someone else will figure it out. It's like, no, you know, if it's not going to be there, if no one's going to do it, because there's a lot of money in keeping things the status quo. I like to come up with solutions and figuring things out, and I'm a seeker of truth. Um, there's no I'm being highly apolitical in this entire guide, and um, you know, I'm not talking about any of the policies that were enacted or cognitive dissonance that people are experiencing today and not masking and all that kind of stuff. That is not what this is about. This is about healing from all this stuff and giving you real tools that you can use to improve yourself so you don't have to be on a drug forever. And you don't have to be living from symptom to symptom. You know, I did that, and I like, man, I had so many symptoms. So let me just check any other questions real here. Um, what viruses do I have? Uh, you know, I want to say it was a combination of EBV and dysbiosis. I acquired EBV from my roommate, sharing a beer, and I was prescribed antibiotics for it. Yeah, it's per month because I'm building this out and giving you more and new tools. So it's not just how to overcome post-viral illness. It's how to do that and how to learn how to enhance yourself beyond that. Like I was not this resourceful before. Like I wasn't born this way. So that it's an ever expanding process. But you're welcome to cancel at any time once you've gotten your value from it. Is there any point in treating SIBO directly? You know, SIBO really works on the oral microbiome, which causes the motility, initiation of motility. I don't, you know, rifaximin and all those other kind of things to try to put it in check is a good adjunct, but it's not addressing the root cause because you're supposed to have bacteria everywhere. You're not sterile. Um, Can you pay via PayPal? I don't know. The system I have, I don't know if it works that way. How often will I be updating the guide? Pretty much every week. um, got tons of things to talk about. Look, I've got a huge database. Before I started doing blogging, I would build out epigenetic databases. Um, so for all this post-viral illness stuff, I've got thousands and thousands of papers that need to be translated appropriately into the layman on here. So this is ever expanding. It's a resource that will like always help you, especially if you love your health and you want to be healthy. All right, guys, I really appreciate your time today. Oops. I really appreciate your time today. Seriously, thank you so much for being here. The discount code will be active for the next 7 days, and I value your feedback. Please, you know, I want to improve as much as I can for you so I can help you. I'm not trying to be a someone to stop you from reaching your goals. I want you to be able to reach your full potential. That's what my biohack is about. Unlock your maximum potential. So thank you guys, and I sincerely appreciate you being here.